We've spent the past several lectures trying to understand how biochemistry works, but there's actually ways to make biochemistry work for you. I remember hearing this saying during my scientific training that the only way for you to build something is to first understand it. So I think all of you are ready to learn how to build with biochemistry, aka synthetic biochemistry. The learning goals will be to first illustrate only some of the biochemical strategies that scientists use to perform something called oligomeric synthesis in the lab, and you will then be evaluating what are the pros and cons of synthetically deriving important biomolecules versus just extracting them from an organism that made it itself. This molecule of the moment is going to be challenging. It's diglycine, a very simple molecule just by looking at it. It's the simplest peptide conceivable. It's just a dimer of glycines, and it's a pretty useful substance. It's a non-toxic compound, and we commonly use it for buffers for poorly soluble proteins. So my question for you is twofold. Consider this diglycine molecule, and I want you to draw out the condensation reaction of a generic amine with a generic carboxylic acid to form a simple amide. What you basically learned in organic chemistry. Now I want you to apply it to this specific molecule and explain why this diglycine might be difficult to synthesize in the laboratory from just a solution of its monomer, which is glycine. On to the main event, you might be aware that biomolecules, proteins especially, have commercial value. All right, think about diabetes. Before people understood the disease, they actually died of starvation shortly after symptoms became severe. And after insulin, a protein was discovered, diabetics had to depend on bovine or porcine insulin, which was costly to extract and highly allergenic. But recombinant technology, which is the process of inserting a valuable gene into, say, a bacterial vector for mass production, completely changed the game. It allowed us to produce enough human insulin to keep our diabetic population alive. And that's just one example of a protein that has commercial value. There are, however, many disadvantages to recombinant technology. Okay, it's not like you give a plasmid to the bacteria and it goes ta-da and starts bleeding insulin for you. There are many practical issues here. First of all, large volumes of bacterial culture are required for mass scale production, and that's going to result in a lot of media waste with every single batch. Furthermore, biology doesn't really do small proteins. The smallest on record, I believe, is 20 residues long. But really, insulin at 51 residues is already near that lower limit of what an organism will transcribe. If you want a 10-mer peptide, this approach isn't viable. Synthetically, recombinant technology also has what us chemists call poor atom economy. What does that mean? Yes, if you give the insulin gene to a bacteria and train it to make it for you, it will make a decent amount of insulin, but it will also make literally every other protein, lipid, whatever it needs to survive, which we really don't want. And we're going to have to then purify all that background and junk out. For every milligram of insulin your bacterial workers are going to make for you, they're going to make grams of junk that you have to sift through. And trust me, there is nothing more painful and tedious and just awful than purifying compounds in the lab. And let's assume we get to that end product easily or simply, where we get the protein, we purify it, and we have our product. Well, it's not ready for the shelves quite yet because we have to consider genetic stability. DNA polymerase has a very small but non-zero error rate. Each nucleobase has a 1 in 10,000 chance or so of getting mutated. So over time, your insulin quality can degrade because future generations of your bacteria might accidentally mess up your protein product. And lastly, this often gets overlooked by inexperienced researchers and lab people. Post-translational modifications, they are a headache, all right? You could give a gene to a bacteria and it can make it, but it could also glycosylate it randomly or in ugly ways that ruin its functionality. Or heck, even make it allergenic. So all of these drawbacks that I listed are a call to action. Is there a way for us to make protein in a test tube reliably and easily rather than outsourcing it to a complex cell with its own emotions and desires that may compete with your own? So this is a chemical problem, and good news, the chemists have indeed figured it out. There are synthetic methodologies for making biomolecules in the lab without having to outsource it to finicky cell culture. Researchers, clinicians, and industries resort to this all the time. I myself did a bunch of it when I was studying to earn my degree. And I'm not just talking proteins. You can even craft a custom gene in the lab, or you can have someone do it for you for a small fee. I want to direct your attention to this screenshot down here from genscript.com. Genscript is kind of like the Walmart of genes. You can get so much from that website. I'm not affiliated with them, but I want to show you that this COVID spike protein was up online not far into 2020, which is quite impressive. And that's useful for scientists who are trying to find ways to combat the disease because they were able to make that spike protein themselves and then do whatever necessary experiments on it. So custom ordering, custom making genes synthetically is totally a thing, and you can do the same for peptides. Biosyn.com is the first website that came to mind. Again, I'm not affiliated with any of these. There's many options for you to choose from, but they sell peptides and proteins. In this screenshot, I'm showing you their page for amyloid beta. This is a supposedly toxic protein that's believed to be implicated in the pathogenesis of Alzheimer's disease. So there's a lot of research interest around understanding how this protein works. You can make it yourself, or you can buy it and do experiments on it. And not all proteins or oligomers need to be so exotic and expensive. If, for example, you're into the cosmetic industry, arginine is a very abundant example of a simple hexameric peptide that's commonly found in wrinkle creams, I believe. And really, the only way for you to make such a short compound in large consumable scales is through organic synthesis. So making oligomers synthetically, whether they be nucleotides or proteins, it's a very profitable and important business. Now, before we get into how scientists make proteins, let's recap how biological protein synthesis works. And I won't go into too much detail here, so I'll just say to start out, first thing you need is a gene. Stick it in your organism, And you often need to add after that some type of inducer in most cases. This is a molecule that will tell the cell to transcribe that gene that you added in. So RNA polymerase will then come in and make an RNA transcript of that gene. The cell will process the RNA transcript. It will deliver that messenger RNA molecule to a ribosome, where it will then be translated. Specifically, the ribosome is going to read the mRNA and grow the protein in the N to C direction. And you'll do this until the cell is just full and brimming with your desired product protein. And then comes the sad part that a lot of people tend to overlook. You have to kill the bacteria that have been faithfully working for you. You need to bust open those cells and harvest the protein out. Then you gotta purify it, do quality control, and then you're able to sell it and get your profit. And then you start all over with a new batch that you had set aside earlier. Over here, I'm going to walk you through how chemical protein synthesis occurs. And in my opinion, I think it's more of a hands-off process. For example, most of it is actually automated by a machine. This here is a picture of what's called a Liberty Blue. This is a model that I used extensively in graduate school, and I must repeat, I'm not affiliated with any of these companies, but if they want to send me a check, they are more than welcome to. And I think she's beautiful. We called her Cynthia in the lab, Cynthia the synthesizer, and I came up with that name, very proud of it. And it's so cool. You have all these glowing blue bottles that you plug into the machine. And in these bottles, you will measure out and dissolve your reagents, plug it in, hit go, and the device will do all the chemistry for you while you go home and sleep. And in the morning, you will have product to work with. And yeah, I'm kind of romanticizing here, making it sound like a black box, but there's actually a lot of sophisticated chemistry that goes on in the reaction vessel that I think you should be aware of. And it's very different from translation. So how does the chemistry inside this machine works? It does not have cells or ribosomes making the protein for you. It does not even have genes even. Instead, we are going to grow our desired peptide off of something called a resin. And I'm showing that over here as this yellow ball, resin. These are porous plastic beads that look like sand. I guess technically they're microplastics, but they're very useful here. Because on the surface of these beads are special amine groups that can react with the C terminus of a protected amino acid, shown over here. The protecting group is shown as the green PG that I've just circled. The C terminus is unprotected, and this nitrogen is also unprotected. So they are free to react and couple and form an amide bond. Now, we have a resin that's not covered in this amine group anymore. It's covered in a covalently attached and protected amino acid. So I want to pause here for a second and ask you, why do you think we protected the N terminus of the amino acid we just clicked on? I'm not going to tell you here because that might give away the answer to the molecule of the moment, but I'll let you ruminate on that, and you can share what you think the answer is when the time's appropriate. Anyway, once we add on that first amino acid, we then need to deprotect the N-terminus of the amino acid we just added on. By doing that and liberating the amine again, we now have a free amine exposed, which will allow us to do the next coupling step, where we're adding in this blue amino acid. And oh, look, it has its own protecting group on that N-terminus. The carboxyl group of it is free, and it's going to react and amidate with the nitrogen that we just deprotected. So fast forwarding on ahead, we now have a dipeptide attached to our resin bead. And we can repeat this process for not an infinite number of times, but several times to get a decently long peptide or protein growing off of our resin bead. And once we are happy and we have made the desired primary sequence, it's time to get the product off of that bead. We are going to do a cleavage reaction, it's called, which is going to slice this bond, make that clearer, and it's going to release the product, and we can easily separate it from the resin and then do additional purifications and whatnot on the good stuff. That is briefly how solid phase peptide synthesis works, SPPS for short. And you might be wondering, well, that's nice. Why do we have to bother doing all of this peptide chemistry on a solid plastic microbead, that resin that I talked about? Well, describing how the machine is set up might answer that question for you. At the heart of the peptide synthesizer is the reaction vessel. Sometimes we call this the RV for short. And the reaction vessel is basically a glorified beaker, but it has valves and openings at the bottom. So you can flush out waste and reagents as you work. But those valves are covered in very fine mesh coverings. The mesh lets liquid and solvent flow through while trapping and keeping the solid phase within the reaction vessel, and by extension, our growing peptide chain. And why is this useful? Well, we have to think about what our reaction solvent's going to be, and that's DMF. Once we put our resin in the mesh-lined reaction vessel, we need to get it suspended in basically liquid amide. DMF stands for dimethyl formamide. Dimethyl. It's a liquid at room temperature, and oh look, it's very chemically similar to the peptide backbone. So it's really good at keeping it dissolved, which is good for reactions. We have our resin swollen up like a sponge, sitting in a solution of dimethyl formamide, and it's ready to do its first coupling step. Let's say that the first amino acid we want to stick on one of our resin beads is alanine. And let's say it can hold 1 millimole of peptide chain on it. We are going to open up a valve of a vial containing a 10-fold excess solution of our protected amino acid alanine. It's going to come down here into the works and enter the reaction vessel that has our resin. So we have an ALA solution with 10 equivalents, and they're going to react with our solid support, with the help of some microwaves, which speeds up the reaction considerably. And when the reaction is done, we will have 1 millimole of alanine attached to the resin, and there's going to be about 9 millimoles of alanine left over that have not reacted. Well, that seems like a waste, right? Not really. I mean, yeah, it is wasteful, but alanine is cheap, and by having that substrate in tenfold excess, we are going to drive this coupling reaction to completion really quickly and really efficiently. I'm talking a 99% yield. Very rare in organic chemistry, by the way. And then that's where the beauty of the solid support comes into play. When the reaction is done, which only takes 4 to 5 minutes, you just open the drain valve, and those other 9 equivalents of alanine and everything else goes down the drain into your waste container. And you have your resin left behind in the RV because it gets blocked by the mesh. There's no awful purification needed between each step. You just rinse off the junk and then add in the next amino acid. Repeats with the next and the next, and you go down the whole alphabet if you need to. This systematic process of couple and then flush, couple and then flush allows us to make really complicated peptides really efficiently. In the next few slides, I'm going to demonstrate how the coupling reactions work. And the overall reaction that I'm showing here over the red line is simply a condensation between an amine, a carboxylic acid, and they will come together to form an amide, which in biology we call a peptide. Everything you see over here in that red box, these are helper molecules, and I will describe their roles very shortly, but for now I want your eyes to glaze over them and instead analyze this reaction that I've shown. And if you look at it, you might realize that there's something wrong. I'm seeing 2 oxygens on one side of the equation and only 1 oxygen over there, for example. 2 hydrogens there, only 1 over there. The equation's not balanced. How do we fix that? Well, you may also remember that whenever you do a condensation reaction, it's called a condensation reaction for a reason. That's because we're going to liberate water, H2O, as a byproduct. So that accounts for the missing atoms, but it doesn't explain why that happened. To understand where that water byproduct comes from, we need to ask ourselves, what is the nucleophile? What is the electrophile? And what is the leaving group? All right, pause the video. Try to come up with the answer yourself before I tell you. Maybe flip back in your notes to the serine protease mechanism, for example, to find that answer. And I will assume you have done that because I will now tell you that the reaction nucleophile is the nitrogen, thanks to its lone pair. That lone pair is going to attack and form a bond with the most accessible electropositive atom in sight. The electrophile will be the carboxylic acid carbon right over there. It's between 2 oxygens, which are just starving it of electrons. So that's the EP, that is what gets attacked. And when we form a bond to that EP, we have to kick something out to make room for it. That would be the hydroxyl group. OH is going to leave as the LV, the leaving group. Being able to identify nucleophiles, electrophiles, leaving groups, it never goes out of style. So that answers that question, but if you remember your organic chemistry, it's going to bring up another one. Remember that OH is a bad leaving group. It doesn't want to leave. So the reaction won't happen as is, not without a little help. And that's where chemistry comes in. In the cell, what biology has learned to do is to activate the carboxylic acid and turn it into a more reactive group. And we do that with transfer RNA. tRNA acylates at that site, and tRNA will become a good leaving group in that case. I always say biology is just applied organic chemistry, right? But we're not going to do this in our solid phase peptide synthesis machine because tRNA is expensive and finicky and hard to make, even harder to isolate. So we need a cheaper molecule. That is where HBTU, or Hib2, comes in. Hib2 is an activator, and what it will do is react with a carboxylic acid and convert it into a good leaving group. Shown over here in the first line is the mechanism for how Hib2 activates a carboxylic acid. The deprotonated form of a carboxylate actually is a little nucleophilic. I know I told you it's electrophilic, but it can be both. The oxygens have lone pairs on them that can attack other more extreme electrophiles. And over here, as shown in the diagram of the Hib2 molecule, we have a carbon that is between three considerably electronegative atoms. So that's even more electrophilic than anything we've ever seen yet so far. We are going to attack and form a bond between here and there, which we see over here. But oh no, we have to break one of the bonds present, and pi bonds are the weakest, so those electrons are going to hop up onto this nitrogen, and then they're going to hop back down and break the bond with the best leaving group available. And if you notice, there's similarities here between this and the serine protease mechanism. I know, your favorite. And we have to ask ourselves, which of these three bonds are we going to break? The answer is very often the one that has the most resonance. And when we do that, breaking the top bond is the most sensible choice. Because the electrons we shove onto that leaving group end up over here. And they're able to delocalize all throughout this highly conjugated system. So that's like a baby leaving group. And hold your skulls in. It also will become a nucleophile. It's kind of like a squatter that you just can't evict. We're going to kick it out, and it's going to say, oh no, I want to come back in. And the oxygen with that negative charge on it is going to be a pretty good nucleophile. And now the roles will reverse.
The carboxylate that has these two nitrogens stuck onto it is really the only electrophile left in town. So we're going to attack, break the pi bonds, take that lone pair, stick it up onto this oxygen, and then it's going to come back down, which we see over here, and we have to break a bond to make room for it. The bond that we end up breaking is going to now be the one between the oxygen and those two like nitrogen bunny ears is what they look like. Because when we do that, we form a very stable urea-like byproduct, which is very stable and does not react, and once it comes off, it stays off forever, leaving us with an activated carboxylic acid. This is the original COO that we had up there, and that bicyclic triple nitrogen thing is attached to the oxygen with a pretty high energy bond, and it wants to leave. This amino acid is now ready to instantly react with an N-terminus and form an amide bond with it. And it will do that with another substitution reaction where we have this nitrogen serving as the nucleophile, and it's going to attack that extremely electrophilic carbon, form a bond with it, and kick off this entire unit. All of that work to give us an amide bond between amino acid 1 and amino acid 2. To recap, we now understand the role of HBTU, Hib2, which activates the carboxylic acid for condensation with an amine. DMF is simply the reaction solvent. What about this DIEA? Diisopropyl ethylamine. I haven't mentioned it yet, and that's because the role it serves is small and indirect, but it's important. Remember how I said that it is the lone pair on our nucleophilic nitrogen N-terminus? That is what makes it reactive. If there's a little bit of acid, a little bit of water present even, the hydrogen from the water can come on and do an acid-base reaction and convert the NH2 into NH3+. There's no lone pair here because the lone pair got covered up with an acidic proton. So it can't do the reaction. That's not good. So to fix that, we add in a special base called DIEA, which is able to take a proton like that and turn it into something like this. This DIEA is going to activate the amine and keep it active. HBTU is going to activate the acid. So they have complementary roles, these two entities. And if you're wondering, well, why don't you just use sodium hydroxide? Well, sodium hydroxide is going to compete with this guy over here, and it's going to destroy your reaction. If you have a hydroxide attack our activated amino acid, you're just going to convert it back to your starting material. And that's not good either. There's a lot of sophisticated chemistry going on here. I know this is extremely challenging, and I think you're doing a great job. My recommendation is to rewind 15 minutes or so and just watch this series of events again, and just keep in mind that all we are doing here is nucleophilic substitutions. And of course, if this is still a hurdle, reach out and let me help you clear it out. So yeah, just watch it another time, watch it slowly, and you got this. Your heads are probably swimming right now, so let's hit pause and look at the forest for the trees and ask ourselves, what are we trying to do here again? We are trying to build a peptide chain cleanly and efficiently off of a solid support, a resin, and we're going to do this one amino acid at a time. We achieve this chemistry by having a solid support covered in a nucleophilic reactive amine group, NH2s. And these NH2s are going to react with an activated amino acid. I'm drawing glycine here, let's say. That's the first amino acid in our sequence. But if you notice, I put a protecting group, PG, on our amino acid. Why did we do that? Remember that we are adding 10-fold excess activated amino acid for every site on the resin support that we're building off from. Let's draw an unprotected glycine nearby to illustrate what might happen. This nitrogen is nucleophilic, and so is this one. Both can compete and react for the same site. Even if we successfully do the first coupling reaction, which might look something like this, there is nothing to stop this nitrogen from continuing the chain and reacting with another glycine. And then that one can react with another glycine, and then you get runaway coupling. You get these long, random, crazy polymers of glycines, let's say, of who knows how many amino acids long, and you've already ruined the reaction before you could complete it. We can stop this unwanted chemistry from happening by, again, adding it on as a protected amino acid. And then, once we wash away all of the unused crap, we can remove the protecting group and then do the next step. So coupling will always be followed by deprotection. And this is what deprotection looks like. Over here, I'm showing you what the protecting group looks like. That protecting group, protecting the nitrogen, is very weak to nucleophilic bases. And the base that we will use to remove it is called piperidine. That is shown over here. It's like a cyclohexane, but we swapped out one of the carbons for a secondary amine. And oh look, it has a nucleophilic lone pair. You might think, oh, it's nucleophilic, it's going to do nucleophilic attack. I mean, well, not exactly. It's really going to do an acid-base reaction. It doesn't look like it, but this hydrogen over here on this tricyclic moiety is kind of acidic. It will get deprotonated by piperidine and form this carbanion, which you can see over here. That's the lone pair left over from that bond. We're able to do this because of resonance stabilization with the rest of this protecting group, which we call FMOC, by the way. However, that lone pair is able to also fall down like this, form a double bond there, and it will break this bond and that causes this bond to also break. And that's going to liberate 3 different products. This whole thing called dibenzylfulvene, a CO2 molecule which just evaporates into the air. I'm just seeing now that this graphic forgot to put an oxygen there. Very silly of them. And most importantly, we are going to get our free amine. Well, almost. There's going to be a negative charge on that nitrogen, which nitrogen doesn't really like. So that negative charge is going to react with the piperidine that we just protonated. And it's going to bring it to a happy NH2 that's neutral. And then we'll restore the piperidine back to its original state over here. And this isn't really crucial, but the dibenzylfulvene and the piperidine will react together into this giant thing that's really easily flushed out. And that chemistry will involve more nucleophilic type reactivity. But the important thing here is piperidine was able to very easily remove that entire FMOC protecting group. Take note that the DIEA from 2 slides ago, this was a base, but it's not nucleophilic. It can't do the reactions that piperidine can do, which is a good thing because that would prevent premature deprotection. It's not nucleophilic, so it can't do a lot of this chemistry because it's sterically hindered. DIEA is a tertiary amine. With lots of bulky groups around it. Those prevent it from getting close to this sterically hindered FMOC group as well. They can't touch each other and do this proton exchange and all the good stuff that follows afterwards. It's complicated, but I think it's really clever. Hopefully by now you can anticipate a pattern emerging where we build our peptide chain by having our solid support and then we couple on the first amino acid. We rinse out the junk, and then we deprotect it so we can couple on the second amino acid. Then we deprotect it, rinse out the junk, and do the third, the fourth, the fifth, until however many amino acids we deem necessary, until we have our peptide complete but chemically attached to this solid support that has been helping us rinse off the junk so we can continue. Once we have our desired peptide, it's time to break it off of our solid support through a reaction known as cleavage. The solid support contains a special dibenzyl linkage that I'm showing over here that's very weak to acid. And the acid that we use to cut the bonds over here between the first amino acid and the resin is TFA, trifluoroacetic acid. It's acetic acid on steroids. TFA is such a strong acid that the amide bond between the peptide that we just made and our resin can become protonated in the following manner. This double bond O can accept a proton from here. This is going to form an oxonium intermediate, which is not stable. Oxygen does not like having a positive charge on it, and the way you get rid of a positive charge is by sucking in electrons. So the closest electrons up for grabs is the pi bond on this C double bond O. It's going to suck those up, and that's going to create an electron hole over here. That electron hole will get filled by the electrons over here, and the bond between that nitrogen and that dibenzene system shown up there by the star. What we're doing is, it's like hot potato. We're moving this positive charge to the first stop, to the second stop, over there. And that's where the positive charge is going to stay. Carbocations are unstable to a degree, but think about resonance. That positive charge is going to be delocalized all throughout both benzene rings, which was going to make it way more stable than the oxonium or anything in between. The last step in the catalytic cycle will be the TFA that has been deprotonated. It is going to take its proton back from the peptide that got protonated. And this is going to create an electronic rearrangement that's ultimately more stable, where the electrons that hopped up onto there will fall back down, and these are going to attack another TFA molecule and deprotonate it, giving us the final product, which is going to be a peptide that ends in an amide bond. A C-terminal cap. You may not have been expecting this. You may recall that peptides have an N-terminus, an amine, and a C-terminus, a carboxylic acid group. And our peptide that we made synthetically doesn't end with that. And that's actually kind of deliberate, because that C-terminus is really easy to react, and it could behave unpredictably in the lab and such. So this amine or this amide at the end makes it a more stable product. But other types of resins out there, when you do this cleavage reaction, it's going to rearrange so that you end it in your expected free acid, like so. But those are a little more difficult to work with, and usually we use the C-terminal amide cap in the lab. TFA does a whole lot more than just liberate the peptide from the resin from which it grew from. I want you to remember that many amino acids have reactive R chains that we need to protect as well. Think about lysine, for example, which I'm kind of showing over here. Lysine has 3 important functional groups: the N-terminus, the C-terminus, and its side chain amine. The side chain of lysine and the N-terminus are both considerably reactive nitrogens. Therefore, we need to protect them to prevent runaway coupling at both of these sites. We can't, however, use the same protecting group. Remember that we use FMOC to cap off the N-terminus to stop that runaway coupling. This is base labile, which means only base will destroy it, like piperidine. If we had FMOC on the N-terminus and over here in the side chain, that's going to create a fork in the road where both this nitrogen and this nitrogen could be the site where the next amino acid attaches. That's not good. So we need to put a different protecting group that is immune to piperidine. And the protecting group we use is BOC. BOC, like that dibenzyl linker that I showed you previously, is acid labile. TFA is going to remove it very easily. The deprotection mechanism is down here. I don't expect you to have it memorized or anything. I just want you to see the similarities and how convenient it is. The BOC protected amine is shown over there. This whole group is BOC. And when we protonate it with a very strong acid, it's going to form an oxonium. And oxoniums are unstable. The oxonium is going to prompt an electronic rearrangement to get this into a more stable configuration. This bond will break, that bond will break, and we're going to have a few other rearrangements occur, and ultimately get carbon dioxide, which will bubble out and clean itself up, and isobutene gas, which is also going to bubble out of the solution because it's very poorly soluble and very volatile. This is going to leave us with our desired free unprotected amine. And hopefully you can see that BOC deep protections are both thermodynamically favorable and entropically favorable. I love a reaction that cleans itself up. It's incredibly convenient. And so TFA is not only going to liberate the peptide, but it's going to help remove all of the protecting groups on the polar and reactive side chains of the peptide that we just made. And if you're curious, you can check this table over here to see all other types of protecting groups that are used for these non-aliphatic amino acids. I want you to know that I don't expect you to learn these by heart. Don't memorize them. Don't memorize their mechanisms. I just want you to be aware that they exist. We have many protecting groups for many different amino acids, but what they all have in common is that they come off with our handy-dandy TFA. How about we now ask ourselves, how efficient is solid phase peptide synthesis overall? What is the overall reaction yield? Now, I told you a few slides back that we can assume each coupling reaction occurs with 99% efficiency, and that is really amazing. What happens, though, when you iterate or repeat a 99% successful reaction with another 99% successful reaction for however many amino acids you have in your sequence? How quickly do those 1% of mistakes accumulate? Said another way, what is the longest peptide that we can feasibly synthesize within the lab before it gets to the point where the yield is so low it's just not even worth trying? You can actually treat this a little bit like a math puzzle and figure it out with pen and paper. And I can show it to you. If we think of that 99% yield as a decimal, 0.99, we can say, all right, if that reaction has a 99% success rate, if we do that reaction 10 times in a row, it's like 0.99 times 0.99 times 0.99, to the power of 10. So 0.99 to the power of 10, what does that equal? I have that punched into my calculator. And that is 0.904. 10 coupling steps, a decamer peptide, we can expect theoretically to form on resin with a 90% success rate. Those other 10% of peptides have some mistakes in them. Maybe an amino acid got skipped, maybe something else happened, who knows what. We will just have to purify those out, but most of the peptide is good and usable. A-. Pretty happy with that. Not many peptides in nature are only 10 amino acids long, though. What does the reaction yield look like for a 40-mer? 40 amino acids long. That's a pretty small protein. But can we make it with solid phase peptide synthesis? Survey says 0.99 to the power of 40 equals 0.669. Said another way, the overall yield is 66.9%. And that's not amazing, okay? When you make a batch of cookies, you like it when all of the cookies are edible, or maybe one or two get a little misshapen. You don't want a third of your tray to end up in the garbage. So that's not ideal, but oatmeal better than no meal, as they say. Let's make it a little bit longer. 70 amino acids long to the power of 70 equals 0.494. At 70 amino acids long, we dip under the 50% mark. At this point, more than half of the peptide we made on that resin has some sort of error in there, and it's not what we want. It needs to be purified out. So the rule of thumb is, what is the yield of solid phase peptide synthesis? Well, it's going to decrease the longer and more complicated your protein is. And generally speaking, solid phase peptide synthesis is only best until somewhere between 40 to 70 amino acids long. Your mileage is going to vary depending on what your primary sequence is. It's totally possible for your growing peptide chains to tangle up on the resin surface and therefore not react nicely. Really, really long peptides might be hard to purify. So as you get closer to 70 amino acids long, it's like, don't hold your breath. It may not be feasible. You may have to do this recombinantly. If you really, really, really want to make a long protein using solid phase peptide synthesis, there are a few elongation strategies that you can use at your disposal. The easiest way to do it is called the native chemical ligation. What this method entails is that you basically divvy up your synthesis job into two batches, where you make the first batch as one unprotected peptide, and then the second half is going to be in your second batch in a second unprotected peptide. But your first peptide, you're going to build it off of a very special resin, a special solid support, so that when you do your cleavage reaction, you get a C-terminal thioester. We've seen thioesters a bunch. Biology uses these as good leaving groups. And you can do something pretty clever if you make sure your second peptide begins with a cysteine. A cysteine is able to do a trans-thioesterification, it's called, and it will perform a nucleophilic attack over there and kick off this R group as a leaving group. So the cysteine side chain, which is strange, is going to form a covalent bond with the backbone of this unprotected peptide 1. And what happens next is that's going to bring the N-terminus into a proximity, the N-terminus from peptide 2, and it's going to basically take the place of the cysteine side chain, and it will do what's called an SN acyl shift, where we're going to move the backbone into the proper position, and we will get our amide bond formed between the first half and the second half, and the cysteine is liberated as it should be. This is very easy. It's a water-safe reaction. It's efficient. The only downside is that if your primary sequence doesn't have a cysteine in it, you really can't do it. There are other methods out there that don't require cysteine, but they're not as easy. Still, nonetheless, this is such a reliable strategy that some people have been able to make proteins that are 300 amino acids long, which is really impressive. Let us now quickly recap the two approaches one could take towards making a protein in the lab. In the natural camp, or the biological camp, these are the major downsides. Small peptides are not viable. If your goal is to make a 10-mer, a 20-mer, don't even bother trying to teach a bacteria to make it for you. It's just going to destroy it before you can harvest it. Another downside is the fact that you need to use a bacterial vector. You will have to design a plasmid, which is work. You will have to convince the bacteria to take it up and incorporate it into its genome, and all sorts of mistakes can happen along the way. Let's say that plasmid gets into the bacterium. Well, once it makes the protein, you need to sift through all the junk, all the other proteins that bacteria makes to find the one that you want. There are tricks out there to make that doable, but it's still an annoying step regardless. And even if the bacteria makes the protein and you're able to have a method that lets you isolate it, the bacterium could do all sorts of unpredictable post-translational modifications to it. It could stick on lipids, it could stick on sugars, and it may not even be consistent in how it does that. So all of that is very bad, and on top of it all, every generation that bacteria multiplies introduces the possibility that a significant mutation could occur to your carefully constructed plasmid containing the protein's synthetic instructions. Very lastly, if you want to make a whole bunch of protein and you want to sell it, good for you. I hope you get lots of profits. Send a check my way. But you need to also consider the waste. Media waste is expensive to process. People don't like it when you dump E. coli down the drains, so you gotta autoclave it. That's going to run up the heating bill, and there's all sorts of price add-ons that you need to consider moving forward. Now, to talk about the downsides for chemical synthesis, I'm not going to say one is better than the other. They just have different handicaps. For chemical synthesis, SPPS, well, large peptides are very hard to make, if not outright impossible. All right, even 200, 300 amino acids isn't a huge protein. If you want to make something really big and complex, you're probably best off doing it in a biological vector. Another downside is that the reagents used to do solid phase peptide synthesis are pretty noxious, and they can even induce people to become allergic to it, as in the scientists doing the reaction. I've done a whole bunch of solid phase peptide synthesis in my career. I haven't grown, an extra head or anything crazy like that. But I've heard stories where people become so sensitive to these pretty nasty chemicals that if they walk into a room with a bottle of Hib2, the thing that activates the amino acid, they can start getting hives on their skin and get breathing problems. They're pretty, nasty things that need to be processed. And that as well is expensive and not good for the environment, dealing with organic waste and all. Another downside of chemical peptide synthesis is that the actual reaction times take longer than the biological scale. I believe a bacterium can translate a protein in 5 seconds, which is astonishing when you consider that one coupling step by solid phase peptide synthesis takes 5 minutes. So we're 60 times slower than a bacterium in a chemical sense. We're chemists, but at the end of the day, I hate to say it, biology does chemistry better than we do. Don't tell my peers that I said that. Anyway, another way that solid phase peptide synthesis may be not useful is, what if you want your protein to have post-translational modifications? Well, if those are something that you're trying to study, or if they have some unique biological effect, you just can't feasibly recreate that at a synthetic scale. Biology has all sorts of precise machinery that's able to do that for you. And scaling is always going to be difficult. Scaling will require you using rather expensive starting materials, and again, it's just going to generate a lot of waste. For every 10 molecules of activated amino acid we throw at our resin, 9 are ultimately ending up in the garbage can. So it's not a clean process, but it does have its uses. They both have their uses. Your job is to understand the pros and cons of both and then decide which way you should go if you ever find yourself in this position of, hey, I need to make proteins. So now that you have the theoretical know-how to make and design your own proteins and genes, I think it's important that you're aware of the power and responsibility that goes along with that knowledge. For a couple of decades now, scientists have been redesigning nature to address important problems in health and industry and the environment. So biochemistry is more and more becoming a technology. That's amazing. However, that means money is getting involved, and money makes everything complicated. I believe it was around the 1980s when a professor by the name of Chakraborty, he engineered a bacterial strain that was able to degrade oil spills. I mean, what a wonderful piece of technology that is. That's something with real environmental and commercial value. And I believe it was the first example of a genetically modified organism. And so his team attempted to patent that technology. And that patent was very heavily contested. I mean, think about it this way. Modern legal doctrine states that organisms aren't property. That's a very slippery slope to go down if people start claiming ownership willy-nilly. But what Chakrabarty's team argued was that we're not trying to patent the organism, but rather the innovation behind it. You know, this strain of bacteria, Pseudomonas putida, is not naturally occurring. And so ultimately, the courts decided that yes, we can't patent organisms, but we can patent GMOs, the technology that makes that organism useful and innovative and unique, and thus paving the way for GMOs in the market. So that seems like a pretty simple rule. You don't own the organism, just the new bits that you stuck into it. All right, well, what about entirely novel organisms? In the 2000s, scientists cobbled together bits and pieces of essential DNA from a variety of bacteria, and they injected them all into an empty cell. And it worked out and started dividing and doing all the things that biology does, making this Mycoplasma laboratorium the first completely synthetic organism. It doesn't occur in nature, just like Pseudomonas putida, but all of its parts were designed from nature. Is that intellectual property? Last I checked, the jury's been out on that. And you can again imagine what a slippery slope that is. I mean, technically, your parents assembled your DNA together. That doesn't mean they own you forever. No way. The more you think about it, really, the more conundrums you can sort of envision. Let's pretend you're a scientist and you're trying to invent a protein to solve a specific problem, like plastic waste. You go to the lab and you make, with solid phase peptide synthesis, your very own enzyme that can eat plastic. And let's say that you're successful. Hooray! Remember a few lectures ago when I mentioned that bacteria Ideonella sacaiensis, that bacterium that was found in a lake outside a water bottle factory that learned how to eat plastic waste by making its own PETase? How does your protein compare to this guy? Is it different enough to be patentable? Or what if you had discovered it, made the patent, and then learned after the fact that this organism had already made this enzyme? Does that mean you own part of that organism, or does that mean you're the first person to have infringed upon the copyright or trademark of a bacterium? See how sticky this is getting? I mean, let's not forget that we don't know what we don't know. Scientists estimate that we've only discovered 10% of all species, and we haven't sequenced the majority of those. What untold biomolecular holy grails are already lurking in the genomes we haven't fully understood yet? I mean, yes, designer proteins and genes can be intellectual property under the current understanding of the law, but it's going to be pretty awkward if we patent something and then learn that another organism already designed it naturally. So I always chafe when I see, like, in marketing terms, like, oh, this product is all natural. This is all natural. That's all natural. The more you study biochemistry, the more you ask yourself, well, what is natural? I don't know, and I don't know how to apply that definition. Does that mean we should stop engineering and optimizing biochemistry until we figure this out? I don't think so. But I also don't think that means we can just stick our heads in the sand and not ask these questions. I don't really have any answers for you to most of the questions I've asked in this slide, but who knows, maybe one day you'll have the answers for me.